The topics we will be covering in this video are gene regulation, some of the terms related with the gene regulation, gene regulatory network, how we implement the gene regulatory network, the random forest algorithm used, how we implemented the code, and finally analyzing the output file. Now we will start with gene regulation. We want genes to be regulated and controlled. Genes are actually made up of DNA, and DNA gives instructions to produce protein in the process of protein synthesis. One important thing to be noted is to decide which gene will be turned on and which will be turned off. This is often known as gene regulation, and there are many ways in which genes can be regulated. As shown in the figure, we have regulatory proteins that can bind to a certain gene region to increase the rate of transcription for transcription enzymes like RNA polymerase. This is the DNA region where the enzyme RNA polymerase actually binds. This is a form of gene regulation. We can think of it like the cells in our brain don't use the same portion of DNA which the stomach cells use for making hydrochloric acid, because there is regulation of DNA to indicate which cells to use and which to not. Now we get familiar with some of the terminologies of gene regulation. The first one is regulatory gene. A regulatory gene is present on DNA and it regulates some other gene. A regulatory sequence is present just above the gene on the DNA. What it means is our DNA looks something like this. This is the gene which we want to express. A regulatory gene is present somewhere over here on the DNA. This regulatory gene secretes some protein molecules which then grabs on over the gene to make the mRNA molecule. Now, transcription is the first step of gene expression in which a particular segment of DNA is copied into RNA by enzyme known as RNA polymerase. RNA polymerase is the builder enzyme that helps in formation of mRNA. Now, the promoter is a region of DNA that initiates transcription of particular gene. Promoters are located near transcription sites on genes on the same strand of DNA. So basically, a promoter is a sequence of DNA where RNA polymerase binds. This is the sequence of DNA, and a promoter is located somewhere over here on the same strand of DNA. The operator is another part of DNA where something called a repressor can bind. A repressor is a DNA binding protein that inhibits expression of one or more genes by binding to the operator. So this is the operator, and a repressor binds somewhere over here on the operator. So when a repressor binds to the operator, it blocks the enzyme RNA polymerase, and so RNA polymerase cannot move further and no RNA can be made. Therefore, no protein can be made. This is the enzyme RNA polymerase which is blocked by the repressor. Now we will see what is the use of a repressor and an operator. So the repressor binds to the operator, which now works as a on-off switch. When there is lactose present in our body, we want enzymes to be made to break down the lactose. If lactose isn't present, there is no need to produce the enzyme, so the repressor binds to the operator and blocks RNA polymerase, which prevents the formation of protein. So the binding of repressor and operator blocks the RNA polymerase to form the mRNA. Now, when the lactose appears, it binds to the repressor. So the repressor detaches from the operator, and so RNA polymerase forms proteins which are enzymes in this case, which helps to break down the lactose. When all the lactose are broken down, the repressor again gets attached to the operator till some other lactose shows up. So through operator and repressor, that is on-off switch, the genes are regulated. Now we see what is a gene regulatory network. A gene regulatory network represents how some genes encode regulatory molecules such as transcription factors or microRNAs for regulating the expression of other genes. For example, this is the DNA strand, here is the promoter, and this is the target gene. We have an input signal over here and the transcription factors over here. So these are the transcription factors that the input signal generates. This is the target gene which gives the output, that is the protein. These transcription factors are special kind of proteins that control the expression of gene. These transcription factors then bind into these sites and control the rate of production of gene. So the input signal regulates the production of other gene, that is, it decides the rate of production. This is known as gene regulatory network. So basically a GRN is a collection of molecular regulators that interact with each other and with other substances to govern expression level of mRNA and proteins. This is an example of gRNA where the nodes represent the gene and the edges represent the regulation or the interaction relationship between the genes, which constitutes a gene regulatory network. Now we will see the implementation of gene regulatory network. We have used the random forest algorithm. Random forest is a tree-based algorithm which involves building several trees and then combining their output to improve the generalization ability of the model. So we first create a data frame from the sample data, a tree partitioning the data frame based on rules into distinct and non-overlapping regions. In regression trees, the splitting decision is based on the minimizing mean squared error. This is an example decision tree where we can see the root nodes, the terminal nodes, and the leaf nodes. The nodes are categorized among them according to the rules of the decision tree. To build the decision trees, we first use the bagging algorithm to create random samples. From the given dataset of yeast, we create a new dataset by leaving some rows from the original sample known as out-of-bag samples, that is OOB samples. Then the model trains on this new dataset. We repeat this process for number of trees we want in our forest. So several trees are grown and final prediction is obtained by averaging or quoting. Now we see how the decision trees are made. So we divide our training data set into X and Y, where X and Y belongs to the real numbers. We plot all the points of training data set on this graph. Now we choose some tree, that is, we split at this point and we divide the x-axis. Now we create some more splits on the x-axis. These splits now define a tree. Suppose the partitions are numbers 1, 2, 3, 4 and 5. So this was our first partition. So this is how the decision tree is actually made. Because x equals to 3 was our first partition, so x greater than 3 is made as the root node. Now for x is lesser than 1, that is the lowest partition, we have to create the leaf node. Leaf node is basically the squared error node. All the nodes shown in the red circles are basically the leaf nodes. Suppose that this is the region R1, that is x lesser than 1. So the squared error for region R1 is calculated as summation of y hat minus yi the whole square. So we choose a y hat for the region R1 such that it minimizes this whole squared error. So suppose we get the value as 0.8, it is the minimum squared error, that is the average of all the y's in the region R1. So suppose this value is 0.8. We similarly calculate values for all the leaf nodes. So we develop the decision tree in this way, which minimizes error in each of the leaf nodes. So this was all about the working of random forest algorithm and how we make decision trees and how we minimize the sum of squared errors. This was the overall construction of a GRN matrix. Now we will see how this GRN matrix is actually constructed. So these are the variables where the input data sample is loaded. We create a data frame out of these samples. This is the n cross n network matrix which is initialized to 0 where n is the number of genes in our sample. So for each of the input genes, we repeat this procedure. So the target gene is the gene which we want to target, and the input gene consists of all other genes other than the target gene. Here X is the array of all the other genes except the target gene, and Y is the array of only target genes. RFR is an object of the random forest regressor algorithm. The parameters we have fed into this algorithm is n estimators are 200. What that means is the number of trees we want in our forest is 200. The OOB score, that is the out of bag score, is said to be true. That means we are using the bagging algorithm and we are discarding some rows of the sample set. Now what this fit function does is it builds a forest from the training set x, y. This forest actually consists of 200 trees. The feature important matrix consists of the final minimized square errors for all the leaf nodes. All the squared errors are fed into the final GRN matrix. We finally convert this network matrix into the CSV file, which is our final output file. After we have generated the network matrix as shown in the code before, this is the formula for calculating the expression of the gene I at time t plus 1. So at time t plus 1, the expression of the gene I is calculated as the summation of all other genes at time t minus 1 and some constant, where the Wij matrix is actually the network matrix that we generated in the code before. So this is how we determine the expression of genes using a gene regulatory network. To demonstrate the code, we have reduced the input size to 20 genes. This is the output CSV file obtained after running the code. This is actually the weight matrix that we used in the formula described earlier to calculate the expression of genes. So the construction of gene regulatory network ends up with the formation of this weight matrix, which is used to calculate the expression of genes at different time intervals.